Hi everyone. Today we are starting the topic of inheritance. Inheritance transcends time. That is, it still occurs today as it did 100 million years ago, regardless of where you live on this planet. Not many people would confuse an elephant with an earthworm, a person with a flower, a tree with a tarantula, or a penguin with a python. However, at one level, the differences between organisms are not as great as you might think. All living things are made from the same building blocks, cells. At the centre of the cell is the nucleus. The nucleus contains chromosomes, and chromosomes are made of protein and DNA. DNA determines the characteristics of all living things. This is a list of famous scientists that helped us to develop our understanding of inheritance to where it is today. We're going to take a journey back in time to learn about each of their contributions. First, let's start with Johannes Friedrich Miescher. He was a Swiss physician and biologist. He was responsible for isolating a previously unknown chemical from the nuclei of dead white blood cells. He called this new chemical nuclein because it was found in the cell nucleus. The name was changed to nucleic acid and eventually deoxyribonucleic acid, which is DNA. Next we have Sir William Henry Bragg and his son Sir William Lawrence Bragg. They were responsible for developing the new science of X-ray crystallography, which is pivotal to developing our understanding of the structure of DNA. Next we have Phoebus Levene. He was a Russian-American biochemist who studied nucleic acids and identified the components of DNA and the arrangement of the sugar, phosphate, and base in a nucleotide. He believed the DNA molecule was too simple to store the genetic code. Little did he realise how wrong he was. Next we have Oswald Avery. He was an American physician and medical researcher that proved Levene wrong. DNA does hold the genetic code. In the 1940s, Erwin Chargaff, an Austrian biochemist, expanded on Levene's work. He made three significant discoveries. The first being that nucleotides are not arranged in the same order in all species. Secondly, in all species, the amounts of adenine and thymine are similar, and the amounts of guanine and cytosine are similar. The amount of adenine plus guanine is always equal to the amount of thymine plus cytosine. This has developed our knowledge of complementary base pairing. Next we have Rosalind Franklin. She was a British scientist who used her skills as an X-ray crystallographer to identify the DNA. She and fellow worker Maurice Wilkins created an X-ray crystallograph of DNA shown on the right-hand side. From the pattern, they deduced that DNA contained rungs like a ladder and had an X shape, which is a pattern consistent with it being a helix. Finally, we come to James Watson and Francis Crick. In 1951, James Watson, an American molecular biologist, attended a lecture in which Rosalind Franklin presented her research. Using the new information about DNA, Watson and his associate Francis Crick refined the 3D models that they had been building. They used what Franklin had found to make their model and received a Nobel Prize for it. The Watson and Crick model of DNA is what we still use today. That's the end of today's lesson.